Good afternoon. I'm Arshad Mahmood from University of Innsbruck, Austria. And today I'm going to share some part of my work that I'm recently doing. And it's transport of self-nanoemulsifying drug delivery systems across the mucus and cellular internalization. In the background, you can see some of the mountains from the Innsbruck that are very famous for skiing. In my presentation, I would give a short introduction and then methods and results that I have up till now and the conclusion. For gene therapy and some of the, or I would say many of the drugs that are particularly related to the anti-cancer classes, they need to be internalized within the cells. Their target sites are present within the cells, either in the nucleus or in the cytoplasm. So one of the major problem or the target for us is to make sufficient concentrations of the formulations or the drug to be present at the target sites. Different strategies that are being used, and the two most common in this are viral and the non-viral categories. Viral sources, they are very efficient, but problem is that they are very immunogenic. The immune system of our body is very active against them, and secondly, their distribution within the body, it is very much more than to our liking. That is the targeting of a particular organ that becomes a problematic with the viral techniques. So for this, the non-viral techniques, they are more being used, or in these days research, they are more being employed in this case. Still, the safety and efficient delivery of this DNA or the other drugs into the targets is a major task in the pharmaceutical research. As we all know, that the oral route of administration is always the preferred one. So in this case, when we use this oral route, then the major problems that we face is rapid enzymatic degradations or the poor cellular uptake. So my aim is to work on this cellular uptake or to increase it. The ongoing research in different parts of the world in this category, they are working on the liposomes. There are polymer-based nanoparticles and the self-nanoemulsifying drug delivery systems. These systems, they have improved bioavailability, permeation enhancement, and they have a protective effect against the enzymatic degradations. So current aim of this study is to investigate snedds as a carrier system for targeted delivery of drugs or genes to the mucosal epithelial cells. This is the one that I will be most discussing in this part. When I started my work, I went for the first formulation of this snedds. Snedds are basically, we describe them as the isotropic mixtures of some oils or surfactants, and they can have some co-solvents or the co-surfactants, and they have the capability that when they come in contact with the aqueous environment, they are, with just a little shake or with just a little turbulence, they are immediately converted into very minute particles, and this particle size then is easily penetrated through the membranes. So basically, I worked on the first formulation, but afterwards, after performing all of the experiments, then to increase its efficiency, I needed to change a formulation, and then I added one more ingredient into this. The only difference that you can feel is the last one, the oleail chloride and tart combination. This was to increase conjugate. So we could clearly see the amide peak, and secondly, there is a shift of the carboxylic group from almost 1800 or 1900 towards the 1700. So these were the conformations for the formation of the bonds. The formulation was characterized by putting them in the phosphate buffer, and the particle size in both of the formulations, it was almost similar, around about 35 nanometers, and the zeta potential values, it slightly came towards the negative when the conjugate was added in the second formulation. So first of all, when we talk about the penetration across the epithelial or the mucoepithelial membranes, then the first part is for the formulation is to pass through the mucus. So for this, I used the intestinal mucus of the pork or a pig, and we cleaned it and then filled it in the silicone tubes. And then we put the formulation on top of it and allowed to incubate it for 4 hours at 37 degrees centigrade. And during this time, the formulation was observed that how much it penetrates within the mucus. And after 4 hours, we observed these segments, like from 1 to 10, each segment is of 2 millimeters. So we could see almost 26 to 32% of the formulation that was penetrating up till the 6 millimeter of the depth within the mucus. So it was considered comparatively good as compared to the other formulations like existing nanoparticles or other liposomes as well. In the second stage, I went for the fluorescent and confocal microscopy. The purpose of this experiment was fluorescent microscopy I used to analyze the toxicity of the formulation, and confocal microscopy to confirm that these SNED particles, they are actually reaching within the cytoplasms or within the intracellular levels. For this, I used CACO2 cells. These are the cancerous cells within the colon that were used. And I grew them on a special glass plates that are used for the confocal microscopy and the fluorescent microscope. And they were grown like 9 to 10 days until the 100% confluence was achieved. And then same pattern was used, that the formulation was applied on it for 4 hours. And then under the conditions of 37 degrees centigrade and 5% carbon dioxide. And afterwards, we used different dyes to analyze or to make the observations. The 3 dyes that are used during these experiments was the Hoechst dye, the propidium iodide, and the Nile red. For the results, under the fluorescent microscope, there was a concentration gradient that was developed, like using control without any formulation, and then increasing concentration from 0.1% to 1%. 1% means 1% of the SNEDs in the buffer. The Hoechst dye, it was it could show the nuclei of these CACO cells, and then the propidium iodide, the PI dye. This is basically it enters into the nuclei when there is a toxic concentration. So we could not see in the control any output of it, then neither in the 0.1% nor in the 0.5%. But 1% of this formulation, it was found to be toxic. So for the future experiments in doing or while treating with these cells, 0.5% concentration, that was the maximum being tolerated by the cells, it was used. Same pattern was used for studying the confocal microscopy, and the entrapment of this formulation within the cells, it was observed. I took the 3 time intervals. Time 1 was 1 hour, time 2 was 2 hours, and time 3 was 4 hours. So this from 1 hour to 4 hour, I could see. And these green parts, these are the nuclei of the cells, or the CACO cells, and the red part is the formulation. Here I used Nile red to have a better observations out of the confocal microscopy. So we can better see it in the 3-dimensional image out of it. The first one is control, where there is no formulation. In time 1, you see that after 1 hour, the formulation starts to reach within the cytoplasms. And as soon as we go to 2 hours or 4 hours, more and more amount of this SNED formulation, it is reaching within the cytoplasm. And so we could say out of this that the SNEDs, they are particularly reaching within the cytoplasm. In the next stage, up till this was just a qualitative pharma study, and then I wanted to quantify that how much amount of the applied formulation it is actually reaching within the cells. So, but here I used a 24 well cell plate, and the same cockatoo cells they were used. The pattern of applying of them of the formulation was same. The one thing that, for example, if one line was considered as a sample and second line was considered as a control, both were treated in the same way in the start, but afterwards, from the control, I allowed all of the formulation to stay there, and it was considered to be the 100%, while I removed the formulation after the specified times from the samples, and then I broke the cells with the Triton X100 and analyzed. And the marker, the fluorescent marker was observed that how much of this enters over here. The results, the first formulation the formulation with which I have started, I could see from 1.3 or 4% to 1.8 or 9% up till the 4 hours of my study period. So this was the amount of this total applied formulation that was actually reaching within the cytoplasm. And then the second stage, when I went for addition of this the conjugate inclusion into the formulation, it resulted in almost 3 to 4-fold increase in total amount. So the black lines, they are for with the tart conjugate, and here we could see that almost 3 to 6, 4.2% of the formulation it is actually reaching within the cytoplasm. So along with this same study, the one additional study that was done is to find out the pathway by which these SNED particles they are entering into the cells. There are different pathways available for the formulation to enter into this, but more prominent are the clathrin-mediated pathway or the Cavalier method-mediated pathway. So I used drugs that are basically inhibitors of this pathway, and to find out that how much there is decrease in amount of the formulation that is reaching within the cells. So indomethacin, it was used for the Cavalier, it was inhibitor of the Cavalier-mediated transport inside the cell, and chlorpromazine HCl, I used it for the clathrin-mediated transports. So in both of the formulations, the pattern was almost same, that indomethacin could block almost 50% of the formulation, like 40 to 50% of the formulation, and the chlorpromazine, it was almost able to block the 60 to 65% of the formulation. So in general, I could say that about 80 to 90% of the formulation, it is entering by two of these pathways. So this is the work that I have done up till now using only the markers. My next part that I will continue after going back from here is to use actual DNA particles and to include into the formulation, and to see how much effective it is for actual the work that I want to do. And secondly, by knowing these pathways that are being followed, I may be able to increase the amount of the formulation that is reaching within the cells by altering some more into the formulation. So conclusion of this study is that SNEDs are promising carrier system for the mucosal epithelial delivery, and they can permeate the mucus gel layer and reach the cytoplasm after being transported by multiple endocytosis pathways, predominantly by the Cavalier-mediated and clathrin-mediated pathways. And combination of SNEDs with the cell-penetrating peptides significantly increase the internalization. Thank you all for patience and listening.